Auzubillahiminash-shaytanir-rajeem. Bismillahir-rahmanir-rahim. Dear students, Assalamu alaikum. This is my second lecture about the different types of fungi that are present in soil. In this lecture, I will be explaining the different types of fungi that are soil-borne, their roles in soil habitat, and the importance of fungi. So we start our lecture with the introduction to fungi. So fungi are basically an important group of eukaryotic microorganisms. There is a very big diversity in fungi, ranging from unicellular to multicellular. The most important character in fungi is that they cannot manufacture their own food. Therefore, they have to depend on the environment, either on a host or on some dead organic matter, from where they can get their own food. Therefore, they are able to produce extracellular lytic enzymes that can dissolve different kind of material. That is why they are one of the most important group present in the soil, and they play a very important role in the different soil ecosystems. Another important character the fungi have is the presence of cell wall. The cell wall in case of fungi cell walls are made of cellulose or chitin. The most important thing is the presence of chitin in the cell walls of fungi. Although there are other materials that are also present in the cell walls of different types of fungi. Another important character is the body of the fungi, which is characteristic of the fungal body. That body consists of thread-like structures. These thread-like structures are known as hyphae. The hyphae together make body of the fungus known as mycelium. This body of this thread-like structure of the fungus help it to sustain their life in complex environment in the soil. Because they are able to grow on some nutrient, by consuming that nutrient, they can produce hyphal structures and then they can travel from that point source to another, to some another point source. And in this way, they can avoid the complex structure of the soil. So that's why soil is a kind of habitat which favor growth of fungi as compared to other microorganisms. So another thing that is important in case when we discuss fungi is their ability to produce huge amount and huge number of spores. Spores are the small structures they make, and these spores are mostly resistant to unfavorable environmental conditions. They are the resting bodies of these fungi. They can sustain unfavorable conditions, and then whenever there are favorable conditions, then these spores can germinate and produce new thallus, from where new mycelia develop, and then so on. And because in the beginning I said that fungi are highly complex, means highly diverse. Uh, there are fungi which are unicellular, which are known as yeast. So all these characters and these things we'll be discussing in detail in coming slides. So now we come to the first slide of uh, this presentation. So as I said, soil is a kind of habitat where fungi are common, and they are habitual to this kind of environment because of their high field growth. Most common groups of fungi are present in soil that we will be discussing in coming slides. There are around 1.5 million species of fungi that have been reported in soil. And then fungi also make different kinds of relations with other microorganisms like algae and cyanobacteria. This relation is known as lichens. Lichens have an important role in soil formation, because lichens have two organisms in combination. One is the fungal partner, that is known as mycobiont, and the other is the green partner, that is either algae or cyanobacteria. This is an autotrophic organism that can manufacture their own food. So this partner is known as phycobiont. So together these organisms grow, and this is a kind of symbiotic or mutualistic relationship. So they grow and play an important role in soil structure and development. These lichens also they are present on rocks where water is available, and they dissolve different kinds of nutrients that are present in rocks. So again, as I said earlier, that because fungi are important soil, so they are one of the most dominant in soil in terms of biomass. A single fungi can grow depending on the availability of nutrient and water. They can grow and grow and grow and produce mycelia and hyphal structures. So they can be enormous in terms of biomass. And then while discussing the importance of fungi, they play an important role in terms of nutrient cycling also, because they are important soil. So they have importance in the nutrient cycling in soil. Another character that I already discussed is that all of them are heterotrophic. So they extract nutrients from the dead organic matter. So in that way, they return many nutrients back and playing an important role in the cycle, especially nitrogen cycle, phosphorus, carbon cycle, etc. And most important is carbon cycle. The mycorrhizal relationship is a kind of relationship that is between a fungus and plant roots. It's a symbiotic relationship between plants and fungi. Uh, in this case, fungus increase the absorptive area for plant. It supports the plants in many ways. So I will be explaining the mycorrhiza in detail in another lecture that I will be recording later. Then another importance of fungi can be they are the most important pathogens of plants up, and most of the diseases caused in plants are because of fungi. They cause up to 80% losses in yields and also in quality of products. Then they have roles in soil ecosystem as predators, parasites, and decomposers. As I already discussed, their role as a decomposer, because they are very important decomposer of wood and organic matter, of course. Then they are predators of smaller organisms. We will be discussing this in coming slides. Then there are soil fungi which has been used in biotechnology for production of different antibiotics like penicillin, cyclosporine, and levastatins. Then there are some fungi which has been used as biological control. For example, Trichoderma species is a well-known mycoparasite. So they are used against other fungi which are parasitic to plants. For example, Trichoderma species has been used against Rhizoctonia solani, which is an important pathogen. Trichoderma is a soil-borne fungi. It is present everywhere. It's a cosmopolitan fungus. It produces a large number of spores, which are known as conidia. The conidia are white or green. The most important character that for which I mention it here is its macroparasitic nature, especially Trichoderma harzianum and Trichoderma viridi have been reported. And another species, Trichoderma gumsii, has also been reported that rolls over the hyphae of other fungi and have some microparasitic characters, like they produce antifungal substances. So now we discuss about some important groups of soil fungi, which include motile fungi. There are motile fungi. Then there are zygomycetes, ascomycetes, and basidiomycetes. These are four important groups of fungi that are present in soil. So we'll be discussing each group in detail in the coming slides. First of all, the motile fungi, as the name indicates, these are the fungi which has at some stage they have some motile partners, motile things in their life cycle. For example, some spores, for example, presence of zoospores that have some kind of motility organs like flagella. So many members of this fungi have already been removed from being true fungi, right? I mean, they are no more considered as true fungi. They are considered as pseudo fungi. For example, true slime molds. It's a group that's known as that's also known as maxomycetes. The maxomycetes or true slime molds are those fungi that don't have their cell walls, so their protoplasts are visible with naked eyes. This group has a big diversity of organisms, from being microscopic to macroscopic. There's a very important species that is used for that is used as an experimental model. For example, the phycomycetes. So these member have flagellated cells in their life cycle. For example, they have swarm cells. These swarm cells can swim in presence of water because of presence of flagella. Then they feed on smaller organisms like smaller spores or bacteria. They show engulfing behavior. So as I discussed earlier, the role of predator. So here you can see these are playing important role in the soil ecosystem as a predator by engulfing the smaller organisms. So then they are also important saprotrophs. Another group of motile organisms are known as oomycetes. Oomycetes include some important plant pathogens such as Pythium and Phytophthora species. Then another important groups, for example, catrids. Catrids are the true fungi and hyphocatrids. Actually, if I divide all these groups like true slime molds, water molds, and hyphocatrids, I can place these three groups separately. That is, true slime molds, water molds, and hyphocatrids can be placed separately in a separate group that is we can call it as pseudo fungi. So they have already been removed from true fungi. However, only the catrids have been placed in true fungi. These catrids are basically parasites of different animals and algae, and there are some saprotrophs which are also present. These saprotrophs use organic matter for their nutrition, especially they can dissolve chitin and cellulose and pollens. Then important group of true fungi is Zygomycota. Zygomycota is a very is a group that contains a huge diversity of fungi, there are remarkable differences in phenotypes, lifestyle, and nutrition in this group of fungi. They have aseptate hyphae. They lack septa. Then some fungi are symbiotic or host dependent, and such fungi cannot be cultured without host. Others are saprophytic. They can use the dead organic matter for the nutrition. Then there are plant symbiotic fungi such as mycorrhiza, especially arbuscular endomycorrhiza. They come from the fungal member of arbuscular endomycorrhiza belongs to glomales. And glomales are mostly placed along with zygomycota, although sometimes it's placed separately than zygomycota. Then there are some animal symbionts like trichomycetes. They are symbiotic in guts of insects and nematodes. And then there are zygomycetes, which can use or proliferates on breads, such as bread molds. And then there are many microparasites which use other fungi as food. Then the next important group of fungi are ascomycetes, yeast and mold. They are also known as sac fungi or cup-shaped fungi. They are known as sac fungi because of sac-like structures they contain. These sac-like structures are present in the fruiting bodies. They contain the sexual spores known as ascospores. These sac-like structures are also known as ascus. As in this figure, it's clear that each ascus contains a number of spores. These spores are sexual spores, and they are also known as ascospores. You can see different numbers of spores. For example, in this case, there are 8 spores in each, and there are different number. So this number vary from 4 to 8. So up to 8 ascospores are present in each sac in case of yeast, in case of ascomycetes. So in this case, this group of fungi is an important member of higher fungi. The mycelia are septate, although these septa contains pores through which the mycelia can move around. So mycelia along with nuclei that can transfer from one part of mycelium to another part. So another thing that I would like to discuss in this slide is the relationship of ascomycetes with deuteromycetes. Deuteromycetes basically contain the asexual forms of fungi. The asexual forms are also known as the anamorphs. So as the soil microbiologists mostly deal with the shapes of fungi which are present in soil, and mostly the fungi present in soil are present in their asexual forms, and these are basically anamorphs. So either their sexual forms are known or not known. So two things can be possible. In some cases, the sexual forms are also known, and if the sexual forms are known, they are separately named, but they are basically teleomorphs. So when we talk about ascomycetes, we are talking about the teleomorphs sometime, because when we talk about ascomycetes, we are talking about ascospores, and ascospores are the characteristics spores of ascomycetes, and ascospores are the sexual spores. So they are present in the sexual forms of the fungi, and those sexual forms are basically known as teleomorphs. But these sexual forms may have their counterparts, their asexual form, that's called anamorphs. And anamorphs are basically the asexual form of the same fungi. These asexual forms have been placed in Deuteromycetes. So here we conclude with this statement that a fungus may be genetically same but may have different phenotypic identification based on sexual or asexual forms. For instance, I would like to give you two examples. One is the Trichoderma. Trichoderma is basically a soil-borne fungus, and Hypocrea is basically another fungus. So these are two forms of the same, genetically same fungus. Hypocrea and Trichoderma are genetically same but morphologically different. This is what I want to tell you. Now, Hypocrea is basically the sexual form of Trichoderma. It contains ascospores. Trichoderma doesn't contain ascospores. It contains asexual spores known as conidia. Now, the two types of spores are different because one are produced by, not by combining nuclei at any stage. So there are no combination of nuclei. That's why some spores are known as asexual spores. For example, the conidia. Trichoderma have conidia, while its counterpart, its sexual counterpart, that's known as Hypocrea, it contains sexual spores. Similarly, there is another example, is Aspergillus and Eurotium. Now, Aspergillus is basically anamorphic form, and Eurotium is its sexual counterpart. So the two types of fungi are are the are genetically same, but they are morphologically different. So Deuteromycetes contain the fungi for which sexual forms may or may not be known. This is very important to know, that all the members of Deuteromycetes are basically anamorphs. Some anamorphs have their teleomorphs known, but some may not be known till now. The majority of Deuteromycetes have their sexual forms in Ascomycetes. So this is very important to know, that Deuteromycetes have their sexual forms, and majority have their sexual forms in Ascomycetes, as I have already given two examples. That's Trichoderma, and then another is as here another is Aspergillus. So however, we all of them are not Ascomycetes. Some belong to Basidiomycetes also. So we will be discussing Basidiomycetes later on in our coming slides. So as I already discussed, that as soil microbiologists are dealing with soil, and soil are present in natural environment, and mostly asexual forms are present in the soil. Sexual forms are found, a bit rarely by soil microbiologists. The most commonly found soil Ascomycetes fungi are Aspergillus species, Trichoderma species, Penicillium species, Fusarium species, Falciparum species. All these fungi are asexual forms, and they are anamorphs. Similarly, there are other members like Coletotrichum and many more examples. Now in this slide, we discuss Ascomycetes as pathogens. Generally, the fungi are opportunistic pathogens. Some of the well-known, notorious pathogens of plants are Coletotrichum species. They infect very big diversity of plants. Similarly, Fusarium species are very dangerous pathogens of plants. They infect all kinds of vegetation. They can cause disease in roots, leaves, and all kinds of disease. They are caused by Fusarium. Similarly, Alternaria species. Then there are many examples of pathogens. Then some well-known pathogens of human animals that belong to Ascomycetes are Candida species. Candida species are very common in human beings. They cause candidiosis. Then histoplasma species are there. They cause histoplasmosis. And then coccidioids and Aspergillus species. So they cause mycosis in humans and animals. They cause skin problems, allergies, and different kinds of problems and diseases in human and animals. Then the Ascomycetes are used in industry. For example, a number of Ascomycetes have been used to produce different types of antibiotics. Some examples are penicillium, produced by penicillin species. Penicillin produced by penicillin species. Then cyclosporine, that's been produced by Tolipocladium species. And then lovastatin, that's produced by Aspergillus species. And when we talk about industry, we can't forget yeast. Yeasts are members of Ascomycetes. And example is brewer's yeast that's very commonly used in production of bread. Then I would like to discuss some features of yeast here. Yeasts are basically unicellular fungi, and when they grow and then they reproduce by dividing the single cell into two. Now, either this division is by budding or by binary fission. Budding means that a small outgrowth develop on the cell, and this outgrowth starts growing, increasing in size, and then makes a new individual. That individual sometimes remains attached and make a kind of chain or group of cells together. Binary fission means that the cells divide into two. And now we discuss another important group of higher fungi. These are basidiomycetes. They actually make club-like bodies, so that's why they are also known as club-shaped spore-bearing fungi. These fungi are also important because they are known for lignin degrading lignin degradation. Lignin is an important cementing material present in wood. Then actomycorrhizal fungi mostly belong to basidiomycetes. Then important character because of which they are known as basidiomycetes is the specialized structure known as basidia. Basidia are these club-shaped bodies on which the spores are produced, and mostly 4 spores are produced on each basidium. Then another important character in basidiomycetes is the presence of 3 types of mycelia, that is primary mycelium, secondary mycelium, and tertiary mycelium. Primary mycelium is monokaryotic. It contains only 1 nucleus. And secondary mycelium are basically dikaryotic. They contain 2 nuclei, and the 2 nuclei are different from each other. And then tertiary mycelium contain the club-shaped bodies, or mushroom-like bodies, on which the basidiospores are produced. Another important character of basidiomycetes is the presence of barrel-shaped septa in mycelium. These barrel-shaped septa have dual pores on each side. And another important character of basidiomycetes is the presence of clamp connection when they divide when the cells divide. During each division, clamps are formed between the 2 cells. So if I conclude all this, so basidiomycetes are the fungi which are having club-shaped bodies on which basidiospores are present, and these club-shaped bodies are known as basidia. Then these basidiomycetes are present in 3 stages. Primary mycelium, secondary mycelium, and tertiary mycelium. They have barrel-shaped septa in their hyphae, and each septum have 2 pores, one from one side and one from other side of the barrel. They make clamp connection when they divide. During their division, clamps are made between 2 cells. So you can readily identify the basidiomycetes on the basis of presence of clamp connections and barrel-shaped septa. So basidiomycetes are important because of their role as a saprotrophic fungi. They grow very well on dead logs and manures such as cow dung, and they also cause kind of problems in turf grasses, such as they cause dry patch and fairy rings because of their underground growth. Then a number of basidiomycetes are important parasites especially in plants. Like Rhizoctonia is an anamorphic form of Thanatophorus, and the Rhizoctonia is a very important pathogen in a huge group of plants, diverse kind of plants, and they cause losses. Then another important group of pathogens like that are known as rust causes problem in diverse plants, especially in cereals. They cause important losses. And rust is caused by Puccinia species belonging to Basidiomycetes. Then another group of pathogens is called smut. Smut disease is caused in plants. They are important in cereals. They belong to Basidiomycetes. Then Basidiomycetes are important. You cannot forget the role of mushrooms in food industry. And, they are rich source of proteins. Now we discuss the habitats of fungi in soil. The fungi are cosmopolitan. So most of the fungi are present in soil. They are present in all types of soil. As I have already discussed this point a little bit, that the hyphal growth best suit the soil environment because they start their growth from point source of nutrient, and then from there it grows to some other in search of some other nutrient source. So their hyphal growth can sustain their growth. I mean, in finding and help them in finding nutrients and fresh sources. And then the places with low nutrients and less moisture are also suitable for these for fungal growth, having more aeration. The fungal abundance and their diversity also change with climate and altitude. There and the areas with where temperature rises contain different types of fungi as compared to the areas where there is low temperatures with high altitudes. Similarly, the harsh conditions promote the fungi to produce some kinds of survival structures. The survival structures are mostly spores. However, there are some fungi which produce dormant structures like sclerotia. These dormant structures help fungi to survive for a longer period of time in unfavorable conditions. Then, as I have already discussed a little bit, that fungi play important role in soil, and they are sometimes major biomass of microorganisms in the soil. The accurate detection of soil, accurate detection of fungi in soil is always a challenge because of complex nature of the soil medium. And then aggregates are different. The difference is in a very minute level, is at a very minute level. Some aggregates are of different shape, some others are of different shape. I mean, there are difference in shapes. Then there's variability in water and nutrient availability. So the microhabitats are created at very short distances. So there are two types of methods through which we can detect the soil fungi. There are direct methods and then there are indirect methods. So when we say direct methods, it means we see or detect the fungi directly, for example, using confocal laser scan microscopy, then using digital imaging and analysis. There are a number of fluorochromes that are available for accurate staining and analysis. Separating these fluorochromes help in separating live and dead fungal hyphae. Then there are methods for hyphae separation, uh, then through which we separate hyphae from soil. And then there are methods for staining. For example, the hyphae can be stained by diazonium blue. The different types of different groups of fungi can be separated, including ascomycetes, pseudomycetes, and zygomycetes, based on their morphological characters. For example, pseudomycetes have very prominent clamps on their mycelium, so they can be separated from other groups. Similarly, zygomycetes are aseptate, so they can readily be identified and separated. So in this way, we can separate the different groups of fungi. Then if we are using a specific type of fungi that is that's a that we're studying in an experiment, so we can detect that fungi in the environment by specially designing a fluorescent level antibodies for that fungi. So we can detect this fungi in the environment. So then there are indirect methods for studying fungi in soil. For example, there are different molecular techniques that in which some specific signature molecules are targeted. Most important molecule is the nucleic acid. Nucleic acid can be extracted from the soil, and then specific primers can be used for detection of fungi. Similarly, enzymes, we can work with enzymes also that are produced by fungi. Fungi can produce extracellular enzymes. These extracellular enzymes are produced in substrates, different substrates. The purpose of producing these extracellular enzymes is to digest the organic matter, to extract energy out of that. Then there are other molecules, for example, phospholipids. Ergosterol is a specific sterol that is solely produced in fungi, and this sterol is rapidly degraded upon death. So this character can be used to analyze the live fungal biomass in soil. So we can extract ergosterol from soil and analyze. That will be a sign of living fungal biomass. And then we can supplement soil with antibiotics or antifungal compounds. This depends on our experimental design, what we want to do actually. So if we want to remove fungi, then we can use antifungal compounds, or if we want to remove bacteria and only we need fungal biomass, then we can use antibiotics. Then we can use glucosamine contents as a signature molecule. This glucosamine is present in the fungal cell walls. It's a building block of chitin. So now, finally, I revise the role, in brief, the role of fungi in soil. The most important role is decomposition. It's an important decomposer in soil. It decomposes litters and woodlogs. It's the most important decomposer of lignin. Lignin is the cementing material of woods. Mushrooms are basidiomycetes that are known for lignin degradations. They have important role in carbon nitrogen cycles. They are able to produce large amounts of exoenzymes or extracellular enzymes. Then they play important role in symbiotic relations in plants. mycorrhizal relationships are something which are very considerable in whenever we talk about plants. Then they make symbiotic relations with other organisms, like they make with algae or blue-green algae. Such relations are known as lichens. Then they have mycoparasitic relations with other fungi. For example, relationship of Trichoderma and Rhizotonia. Such relations have been explored frequently to search the biological control agents. Then the higher fungi have some role by feeding on bacteria. For example, pleurotus is a mushroom that attack on colonies of bacteria. Pleurotus also feed on yeast, so it's a kind of predator role that it is playing. Then the most important pathogens of plants are belonging to fungi. The most important diseases of plants like root rot of plant, damping of young seedlings, and fruit rot and rust diseases, all these pathogens belong to fungi. So thank you very much for your concentration. If you have any question, you can just put your question on KCMS, so I will answer your question. Thanks.